I would like to first acknowledge the Yugara people and the Turrbal people on the lands which we meet today, and I pay my respects to all First Nations peoples past, present and emerging. But a bit of background on Taronga first, and this image highlights our key roles and responsibilities. And we are actually a New South Wales government entity, and we have legislated responsibilities in these areas of conservation, of research and education. And the work I'm talking about today falls into our conservation action pillar over there at around 3 o'clock. I also wanted to draw attention to our commitment to country pillar. And this is to commit to and embed a way of working that includes and respects Indigenous peoples, place and culture. And this is something that we have to do in our programs, and I will come back to that towards the end of this talk and how that relates to some of the science work I'm talking about. Next slide, please. Very quickly, within our science team, we have 4 broad areas of focus, but obviously we have to undertake research with a multidisciplinary approach, and we work with a whole lot of different organisations to do this. So you'll just be seeing a very small slice of the work that we do. Next slide, thanks. And we obviously work with a range of species, and I've got a bit of a snapshot here on some of the science-based programs that we work on, and they are arranged by animal taxon, but of course the work we need to do needs to be across ecosystem and of course landscape scales. And I've got a few examples, 3 that I've pulled out. So one is more of an international example on the work in the reproductive sciences with the Sumatran rhino. We'll also touch on some work with Great Barrier Reef corals, which is a program initiated by our wonderful facilitator here, and Mary Hagedorn from the Smithsonian Institution in the US. And lastly, some work with some of the amphibian work that we've been doing, and this relates and I guess aligns to a lot of the breed for release programs that we've been involved in. Next slide, thanks. And so regardless of whether we're working with managed populations in zoos, for example, or in the wild, we have these unified goals and outcomes, and we're obviously trying to assess and to maximise genetic diversity so these populations can adapt to change. We want to promote that resilience and capacity to adapt to change. And then the outcome there is maximised animal fitness. And this could be as part of translocation programs within the wild, so wild-to-wild movement of populations, or other examples that are there on the slide. Next slide, thanks. And you can just click through the different animations on there, please, just to the end of that. Thank you. And so what this first example is an example of is with the Sumatran rhino. And this is certainly an example of a species where we've used a lot of reproductive tools to help support it. And it's certainly not an example of where we want to be with any species, but it's an example of what we can generate from studying this species in a managed setting and use that knowledge to support the populations in the wild. And it has to be said that the outlook for the Sumatran rhino is quite dire. There's probably 30 to 40 animals left in the wild. It's in a response to obviously a multitude of threats. And the solitary nature of this species combined with the loss of its connectivity and its habitat has resulted in animals not coming together for breeding. And what we see in a lot of different species, mammals primarily, is that we get the development of pathology in the females in their reproductive tract when they don't undergo those regular cycles of pregnancy and lactation. And so there's a population of 8 animals at this particular sanctuary in Sumatra at Wakambas National Park. And that video at the top there that I was playing before, or that was being played before, just shows one of the animals from this founder population going out into the forest. And it would do this every day, doing what a wild rhino should be doing. But the rhinos do come back into the sanctuary at night for care and for protection from poaching. Next slide, thanks. So we use a lot of tools, as I've mentioned, and sorry about that. And one of the tools is hormone monitoring, and we need to establish what's normal for these animals, and we can do that using a variety of biological samples. Next slide, please. And we also use a technique called ultrasound, and you can flip through to the end of the slide, thank you. And what you see here is Ibu Gandis from the Sumatran Rhino Sanctuary ultrasounding one of the female rhinos there. This technique has generated so much knowledge about the reproductive biology of the species, and it's also really important to time natural breedings for the species, so enhancing natural reproduction. It's very important for this species of rhino because they are solitary in nature, and if the females introduce the male at a time in her cycle when she's not receptive, there can be serious injury, aggression, and even death of either animal. So the staff monitor the size of the follicle, and you can see that middle image on the right there. That's a follicle that is very close to ovulation. And so the timings are made based on this information. Of course, we can also use ultrasound to diagnose pregnancy and make sure that pregnancy is healthy. Next slide, thanks. And that particular female did conceive, and she gave birth to a calf this year. It was her first calf, and she's now 21, and that's quite a sort of an older age to give birth for the first time for a rhino, and it's a very significant birth for this population because it's new genetic diversity that would have probably been lost if these tools had not been available to us. And on this slide, I just also wanted to call out the fact that, the reproductive sciences is only one very small component of efforts to conserve this species. And that 360-degree approach is vital here, as it is in all the programs that we've been hearing about at this conference. And there's other initiatives that are at play. Obviously, addressing the root cause of the species decline is priority there. And there's a lot of community and community-led activities in that space. Next slide, thanks. Cell. But we will talk about cells next. So the technologies I've just mentioned all support natural breeding, so as you can see, but one of the cornerstones of our toolkit, I guess, is biobanking and cell cryopreservation. And we can actually save genes that are crucial for survival into perpetuity. And so we are able to collect these cells and store them in tanks like you can see on the image here at a very cold temperature. So they stay alive and can be thawed out anytime into the future. They're analogous to plant seed banks that I'm sure many of you have heard about, but the work in animal species is a little further behind. Next slide, thank you. There are a lot of different ways to collect the cells, and just a very, very quick snapshot here. In the Sumatran rhino, obviously a lot more challenging, but we do piggyback this on to health assessments for the animals when they're anaesthetised. And that, if you could play the two videos there, thanks. In the middle there, that's the collection of and I'm talking about sperm cells in this case. That's where we've done the most biobanking work. And this is a critically endangered northern corroboree frog. And this frog has received a very small dose of hormone. It's the same hormone it would release when it's breeding a female. And what that hormone does is it induces sperm release, and then we can go and collect it in the urine of the frog. It looks a bit spiky there, but that is a capillary tube, and it's inserted only about 1 or 2 millimetres into the frog. And we've done this with hundreds and obviously thousands of threatened frogs worldwide without any complications. So it's a safe technique, and it allows us to get the cells and then process them for cryopreservation. And then if you could click on the right video there, thank you. And this is just probably the most straightforward process where we're collecting egg sperm bundles from wild Great Barrier Reef corals. And you can see it's just a matter of scooping them off the surface of the seawater there during that spawning window, which is a very small window for each species, but is something we take opportunity to collect as many cells as we can. And just in the bottom there, we've got John Daly and Rebecca Hobbs, also from Taronga, who have been working on all of these programs, and together we've developed mobile preservation equipment, so we can go anywhere in the field and apply these technologies. Next slide, thanks. And so just to go over to the corals, you've heard a bit about the methods and the desired goals of securing that genetic diversity, but there's just a few key things I'd like to sort of draw out. So the work that's been going on with the biobanking started in 2011, and as we've gone further into I guess the time pressures for the Great Barrier Reef, we've been focusing more and more on heat-tolerant populations and colonies that have heat-tolerant or have traits conducive to survival. And if you click on that video just on the bottom left there, you can see some cells after they've been frozen and thawed. And in the middle there, we've got an IVF set up. And I've got a comment there on the bottom of the image. You may not see it in the small font, but we're looking to run these IVF technologies at scale. And it was mentioned in a previous presentation that that we can't develop these technologies if they're not accessible to the practitioners at the end of the day. So end users like reef managers, we have to make sure our techniques are effective but also practical. The other really important point I'd like to pull out, and it's on the slide there, is the extensive and comprehensive consultation that is needed with First Nations peoples when we're doing this work. We work with colonies up on the Great Barrier Reef that are brought into an aquarium setting temporarily. So for the most part, the majority of coral colonies go back out into the reef after they've spawned. But the cells that we're freezing, we obviously are taking them off sea country and moving them to land country where our cryodiversity banks are established. And those facilities are specialised so that the cells are monitored there 24/7. But there's obviously a lot of consultation that is needed, free and prior informed consent. There are programs that are underway where there are traditional owner-led biobanking plans, and so I'm hoping that in the future there could be a First Nations people talking about their experiences with this work and their activity in that place. And next slide, please. And this is my last one. I know I'm over time, so Burke, thank you for not getting up. So yeah, the last slide is just on the frog work, and very, very quickly, the key points here that biobanking and assisted fertilisation technologies have long been recognised as a high priority tool, a conservation tool for amphibians. And there's initiatives and programs in federal government documents, in United Nations IUCN documents. We have started some work with some of our listed amphibian species, including the northern burralong frog. And if you can click on the video just on the bottom left there, thank you. And this is a population of frogs that came to Taronga in 2020 in response to the extended drought and the bushfires. That population was wiped out in the wild. We had 60 animals approximately that came into care. We know that this species lives only for a short time in the wild, so 3 to 4 years, and the offspring of these frogs, they're bred naturally, will be translocated back into the habitat next year because conditions have improved, as you would imagine. But we were able to also bank sperm from all those founder males as well as an insurance policy, because there are some that haven't bred yet, and who knows if they will naturally breed before they pass on. So it's a very, very powerful tool, but it's certainly a tool that is useless unless there are efforts that are addressing the root causes of these declines. So thank you very much, and I will, the next slide please. There are obviously a lot of organisations that we work with and funding agencies, and so I thank them all and thank you very much for listening.